Hello guys, welcome to my YouTube channel. So today we are going to discuss the structure and some properties of the most common plasmid vector, which is pBR322. Now, as you can see, the name itself is a combination of some letters and numbers. So let us understand what does the name signify. So here in the name pBR322, this letter P stands for plasmid. That means it signifies that this is a plasmid vector that we are talking about. Then BR, these two letters together stand for the name of the scientist who first discovered this vector, which in this case B stands for Bolivar and R stands for another scientist, Rodriguez. Next, 322. These three numbers distinguishes this plasmid from the other plasmid that were developed in the same lab. So as you can see on the left hand side, this is the entire circular structure of a pBR322 plasmid vector. Now let us see some properties of pBR322. So the first property is that this plasmid vector is very small in size, of the order of 4361 base pair. Now due to this extreme small in size, it is widely used in recombinant DNA technology for generating recombinant DNAs. Now this plasmid vector pBR322 is actually synthesized from three different plasmids. Okay, we will find out now what are the three different plasmids. So out of this total length of 4361 base pair, from the first fragment till 1763 base pair, this fragment is made up of from the plasmid PSC101, which has tetracycline resistance gene or tetar gene. So on the left hand side you will find this is where the first base pair position starts from the pBR322, and from here up to this position is the 1763th base pair. So all the three base pairs from 1 to 1763 base pair of the pBR322 plasmid vector has been derived from PSC101. So this plasmid has been used to make the first 1764 base pairs of the plasmid vector pBR322. Next, the fragment from 1764 to 3147 base pair. So this fragment has been derived from another plasmid vector, which is PMB101. So this is the second plasmid which has been used to construct the plasmid vector pBR322. First was PSC101, next was PMB1. Alright, so if you observe on the left hand side here, from 1764 base pair till 3147 base pair, so up to here this fragment was derived from the second plasmid, which is PMB1. Alright, and the last fragment from 3148 base pair to 4361 base pair, so this fragment has been derived from a third plasmid, which is in this case PRSF2124, and this fragment also carries the ampicillin resistance gene. So on the left hand side, if you observe from the 3147 base pair till the end, so this entire fragment has been derived from another plasmid, which is PRSF2124. Alright, so now all total three different plasmids have been used. Actually, fragments from three different plasmids have been used to construct the synthetic plasmid vector pBR322. Now, one thing you remember, that the fragment which has been derived from PSC101, this fragment has the tetracycline resistance gene, and the fragment which has been derived from the plasmid PRSF2124, this fragment has the ampicillin resistance gene. The fragment which has been derived from the second plasmid PMB1, this fragment has the origin. Okay, so ori sequence or the origin of replication sequence has been derived derived from the plasmid PMB1. Alright, now there are actually 40 restriction sites all over in total present in the entire pBr322 plasmid, out of which 6 are present within the ampicillin resistant gene. So 6 are present in this position in the ampicillin resistant gene, 11 are present in the TET-R gene, and 2 are present in the TET-R gene promoter. So these are some of the properties of pBr322, and these properties justify why this plasmid vector has been used extensively in genetic engineering. So once again, if you look at the structure of pBr322, you will notice that there are some famous restriction enzyme sequences which I will like to show. For example, in the TET-R gene, this HIN3 sequence restriction site is present for HIN3 restriction enzyme, BAMH1 restriction site is present, SAL1 restriction site is present. These three restriction sites are very much useful in genetic engineering. In ampicillin resistant gene, the most famous and useful restriction site is PST1, which is present, and EcoR1 site is also present, but it is not present on any of the antibiotic resistant gene. It is present outside those sequences, and here lies the origin of replication or sequence. Alright, so this is more or less the structure of the plasmid vector pBr322.